Hi, my name is Erica Hicks, and our group will be discussing bioenergetics. The specific example that we chose is energy coupling in a biological system, discussing metabolism and the production of energy in the form of glucose and ATP. We will discuss both of these important processes. Our group members are myself, Erica, Pete Rivera, and Chelsea Seiler. So, what is energy coupling in a biological system? Before we actually get into our specific examples of the breaking down of glucose and ATP production, we will talk about energy coupling. It is a process in which one reaction that requires energy is dependent on another in order to happen within a living organism. So the processes are dependent on each other. Some steps in the process actually release energy while others are absorbing it. We will discuss how this happens. One example of this process is how the system is used in human metabolism. To give you an example, metabolism is composed of many chemical processes that start with one particular molecule that leads to the production of others. And we will discuss how glucose, which is produced by plants and consumed by animals, is used to create adenosine triphosphate, or the energy molecule that's otherwise referred to as ATP. ATP synthesis is dependent on glucose. When humans consume their nutrients, especially items like carbohydrates, this polysaccharide is taken up by the cells. But glucose is too large of a molecule and it must be broken down. So during this process of glycolysis, glucose is broken down. Later on, 2-pyruvate are made. This process leads then to cellular respiration, where ATP is a product. So in cellular respiration, which is driven by glucose, you must have the presence of oxygen to actually generate ATP. So it's an aerobic cycle. This occurs on the membrane of the mitochondria along the electron transport chain. The chemical formula below, it demonstrates the reactants on the left, C6H12O6, which is glucose, and then the presence of oxygen, 6O2, and it yields 6 carbon dioxides, 6 water molecules, and then ATP energy. We'll discuss this further in the next slide. So if we look at the location of where glycolysis is taking place and where ATP production is taking place, you can see it here in this diagram. So these are the metabolic pathways inside of the human cells, and glycolysis is actually broken down in the cytoplasm, where it produces this pyruvate and then ATP is formed. The electrons, which are carried by NADH, they also are present in the mitochondria. The mitochondrial membrane will use this glucose, and it also takes the electrons that are along the membrane of the mitochondria, and it produces ATP after oxidative phosphorylation, which is important to note because it's the presence of oxygen that causes this large amount of ATP to be produced. Another important point is that ATP itself is not actually what is generating the energy production. It is the process of hydrolysis. So whenever the water molecule breaks this ATP down into ADP, adenosine diphosphate, that is actually where the energy gets released as the third phosphate bond is broken off. A concept check that we would like to present to students to help them recap what they've learned so far is listing the reactants and products in the formula for cell respiration. So they would need to write down the reactants on the left and the products on the right. Now that you have written it down, you would need to explain the formula in a sentence format, not the chemical formula format, and then give real life examples to a partner. Ask each other how this is an important metabolic process and how it applies to energy coupling in a biological system.
Hi, my name is Chelsea Seiler, and I'll be continuing on with this presentation. To start, I'd like to discuss the exergonic breakdown of glucose and how it powers the synthesis of ATP. Glycolysis is part 1 of ATP synthesis, and it has this familiar equation. Glucose plus oxygen yields carbon dioxide and water. This reaction is going to release 686 kilocalories per mole of energy. ATP is synthesized through a series of steps within cells pictured at the right and is coupled with the reaction above. The reason that cells do this is because they need to harness energy in the form of ATP because ATP is a usable form of chemical energy. So why is energy coupling necessary for life? Because biological systems have to do work. That work could be chemical work or mechanical work or transport work or whatever have you, but they all must do work to survive. Some reactions that biological systems have to do require energy input to occur, and we call those endergonic reactions, while others are going to release energy as they occur, and we call those exergonic. Cells are going to use the exergonic reactions that release energy to power the endergonic ones that need energy input. And one of the best examples of this is the hydrolysis of ATP, which is used to power many, many, many biological reactions. So let's take a step back for a second and let's practice. The synthesis of ATP is described in the following reaction below. ADP plus a phosphate yields ATP and water. This is known as dehydration synthesis. And this reaction is going to require an energy input of 30.5 kilojoules per mole. Now, using the table at the right, determine which of the following reactions could be coupled with the synthesis of ATP to make that overall reaction series exergonic. So to sum up, the goal is that you want that delta G to be negative. So you're going to do that by adding the 30.5 with the totals over here to see which ones make a negative number, and those ones will be exergonic. Moving on, let's discuss how the hydrolysis of ATP powers those cellular processes. So ATP powers reactions in cells, not just through simple hydrolysis, but through phosphorylated intermediates. And this is usually a two-step process. It's not always just phosphoryl groups that are used for this. Sometimes it's pyrophosphoryl groups or adenyl groups as well. But I'll be discussing the phosphoryl group intermediate just to simplify things. But first, I'd like to take a closer look at the ATP molecule itself, so I'll direct your attention over here. This is an ATP molecule, and you'll notice that these three phosphates over here, each one of them has at least one negative charge. And negative charges don't like to hang out next to one another. They repel each other, which makes this molecule kind of a coiled spring that contains lots of energy. So when you put ATP through hydrolysis, you are breaking off one of these phosphates, leaving a DP and a phosphate molecule in its wake. So how does that couple with other reactions? Well, let's say, for example, you want to put these two random molecules together to make this structure. But that particular reaction synthesis requires an input of, let's say, 3 kilocalories per mole. ATP is going to come in, and instead of going directly from reactants to products, because remember, this is currently an endergonic reaction, ATP is going to come in and it's going to donate a phosphoryl group to one of those reactants. So instead of going straight to the product you want, you're going to go to this intermediate step. So you're going to have this plus that. Now, the reason that this is beneficial is because this intermediate here is less stable than the original reactant molecule. And that's important because when a molecule becomes less stable, it doesn't like to stay like that. So by destabilizing this molecule, you've made it more likely to bond with the other reactant molecule, producing what you wanted in the first place. And yes, you are going to get rid of that phosphoryl group in the process. So to sum up, I would like to look at a couple of more specific examples On the left here, you see an uncoupled reaction, glucose plus fructose yields sucrose. This would be representative of our endergonic reaction that requires a coupling. Down on the bottom, you'll see the coupled reaction, glucose plus ATP is going to yield glucose bonded to a phosphoryl group plus ADP. So that step is when the ATP hydrolysis takes place. Then down here in step 2, we have that glucose phosphate molecule plus fructose yielding sucrose and that lone phosphate. So we see that phosphate or phosphoryl group or whatever it happened to be left and yielded the product that was originally wanted. On the right here, we see a phosphorylated intermediate in protein synthesis on the top and a pyrophosphorylated intermediate in polysaccharide synthesis on the bottom to just give a better example of the specific structure of those molecules.
To continue our understanding of energy coupling, we can look to a vital system in our bodies essential to homeostasis. This system is a sodium-potassium pump. The process of energy coupling enables this system to function regulating our nerve cells, kidney functions, heart contraction, and blood pressure. It is important to understand that endergonic reactions of cellular processes enables coupling. Furthermore, ATP hydrolysis enables cells to couple the exergonic reaction produced. Energy coupling is not only important to the sodium-potassium pump. Our nerve cells require transmembrane ion pumps, which also use energy coupling to perform their actions. In the case of transmembrane ion pumps, the energy produced by ATP is used by nerve cells to pump ions across the cell membrane, thus generating action potential to trigger a response. In regards to the sodium-potassium pump, ATP hydrolysis enables phosphorylation to occur. This process leads to the transfer of gamma phosphates to the protein pump. The pump then gains free energy from this process, allowing for the release of 3 sodium ions while taking in 2 potassium ions as seen in the image on the slide. Thus, the donation of free energy to the pump allows phosphorylation to enact the endergonic reaction. Now, to further discuss the topic of free energy change represents the difference between the final state and initial state of free energy. What does that mean? We have to look at this as the ability to do work and inability to do work. Let me further explain. When cells have an enormous amount of energy, it becomes less stable as it needs to release the excess energy created. At the same time, the higher the energy, the more capacity there is to perform work, as already stated. Let's consider the picture to the right. The diver on top of a platform has a greater capacity to do work due to the gravity. Molecules created by concentrated release of a dye are able to disperse and cover more area. In a chemical reaction, a molecule can be broken down into simpler form. So systems with a high free energy want to become more stable, thus release free energy as in the process of ATP hydrolysis. Spontaneous events result in a free energy change that is negative. How is this so? Think about the diver jumping off the diving board as a spontaneous event. Initially, the capacity to do work is there, but once he hits the water, this capacity is reduced. Thus, as depicted on the previous slide, free energy change is calculated by the difference between the final state and initial state of free energy. Since the initial state of free energy has a higher capacity to do work, the end result will be negative after the energy has been exerted. Lastly, exergonic reactions occur spontaneously and enable the release of free energy. Endergonic reactions, on the other hand, absorb free energy, as in the process of the sodium-potassium pump in the process of phosphorylation. Now, let us practice what we have learned. Look at the slide and match the events to the appropriate reactions. I will give you a few seconds to consider your answers. Now let us review. In exergonic reactions, free energy is released, while in endergonic reactions, free energy is absorbed. Exergonic reactions are spontaneous, while endergonic reactions are non-spontaneous. Finally, exergonic reactions are considered negative, while endergonic reactions are considered positive. Remember, free energy source is the energy that is ultimately released from the breakage of phosphoryl and hydride bonds, the same bonds seen in ATP. In conclusion, we have covered energy coupling in a biological system, ATP synthesis and its reliance on glucose, ATP and glucose interdependence, exergonic breakdown of glucose, why is energy coupling necessary for life, hydrolysis of ATP, coupled and uncoupled reactions, energy coupling, sodium-potassium pumps, free energy change, and exergonic and endergonic reactions comparison. Thank you so much for listening to our presentation. Have a good night.